Hi, everyone, and thank you for joining us today. The European Association for Cancer Research is very pleased to introduce you in this virtual event to the CGI-Clinics project that we partner in. This webinar will give you the opportunity to experience the next generation of genomic analysis with the Cancer Genome Interpreter. This session introduces the one-stop shop platform developed by the CGI-Clinics project, which leverages advanced machine learning to transform cancer mutations into actionable clinical insights. Today, we invited 3 speakers to discuss their research and how it contributes to the CGI-Clinics project. We will have a total of 4 short presentations between these 3 speakers, and I'm delighted to welcome Dr. Nuria López-Bigas, Dr. Santiago Demajo, and Dr. Loic Verlingue. We also welcome Olivia Tort, who's the project manager and will help us with the Q&A session at the end of the webinar. We are aiming for 20-25 minutes Q&A, where participants will be able to type questions throughout, like you can type the questions throughout the talk, and then at the end, we'll give you the opportunity to ask your questions live and interact with the panel. We encourage you to upvote questions as well, if you'd like them to be answered. And we would love for this webinar to be interactive. So please do open your mics if you're available when you're invited to do so. The Q&A box is at the bottom of the screen, so if you can't find it, it might be hiding under the More button, which is represented by the 3 dots. If you click on that button, the Q&A box will pop up. You can add questions at any time during the panel discussion. Just please use the following format. If you want to address the question to a certain speaker, please name the speaker before you type in the question. Otherwise, you can just type in the question and the speaker that feels that most confident to answer it will just answer it. And remember, there are no wrong questions, and we encourage you to ask even if you think it seems simple. You can also ask anonymously if you really want to, and I'll make sure your question gets answered, and I'll read it out loud for you. So I'll now introduce the first speaker, Nuria López-Bigas, an ICREA research professor at the Institute for Research in Biomedicine in Barcelona. Her lab combines strong expertise in biology and bioinformatics, and it is focused on the study of cancer. They have made major contributions in the identification of cancer genes and cancer driver mutations across tumor types, and is understanding the mutational process leading to the accumulation of mutations in cells. Nuria was elected member of the European Molecular Biology Organization in 2016, fellow of the International Society of Computational Biology in 2021, and member of the Spanish Royal Society of Sciences in 2024. Then in 2025, she received the I'm hoping I'm pronouncing this right, so I apologize in advance. Genome, the first award in biomedical research. She currently leads the European project CGI-Clinics, which joins cancer hospitals across Europe to develop and implement a data-driven cancer genome interpretation for clinical decisions making. She calls the Cancer Grant Challenges project prominent as well. So Nuria, please take the stage and share your presentation with us.
Okay, yeah, thanks, Alexandra, for the introduction. I'm gonna focus my part of the talk on the science behind the Cancer Genome Interpreter, and the other speakers will talk more about the tool itself and the platform itself and how to use it or how it's being used in a specific place. So just the motivation is this challenge. So at the moment of sequencing either a whole genome, whole exome, or even a panel of genes in a tumor or a liquid biopsy, you are faced with the challenge of interpreting the genome alterations that you identify and what information they may have for the decision of the treatment or others. And the first challenge that we have is that the relevant information, the knowledge that is about these variants that may be useful is scattered in different places. And for example, you have to identify which mutations are there and go to genome mutation databases, annotate the consequence of the mutation, biomarkers databases, scientific articles, abstracts, clinical trials, case reports, and knowledge from experts that are around you. And getting all this information, it's time consuming, it's often unsystematic. So one way to address this is to try to put all the information together in a platform in an organized way and make sure that all this information is available at the time of interpreting. This is one of the things that Cancer Genome Interpreter does, and this is one of the things that we are presenting today. And you will, so for example, the platform at the moment has mutations and biomarkers databases, expert knowledge, and also access to virtual molecular tumor boards. You will hear more about all this by Santi in a few minutes. But I want to focus in the second challenge, that is that even if we put all the relevant knowledge that is scattered around in all these different places into a platform, we still ignore the significance of most tumor mutations. And one way of seeing it would be with this map. This is a real map from 1525 of the world. At that moment, the east coast of America was well known, but the rest of America was basically unknown. And many maps at this time were basically inventing what was in the other side. And this is a very honest map. That's why it's interesting. Recognizing that there is a lot that was not known and it had to be discovered. So this is a bit the same thing that we are thinking for cancer genomes, right? For example, we don't have to think that we know all cancer genes, that we know all cancer mutations, and see if we can discover them in a systematic way. And that's why, as part of the things that we have done in the lab, and also an important part of Cancer Genome Interpreter is this automatic learning platform to learn from thousands of tumor genomes. So this is data-driven methods that from the sequences of thousands of tumors, we would like to learn directly, for example, which genes are cancer genes. How we can identify those, we realized early on that identifying signals of positive selection in the pattern of tumor mutations in a cohort is a very effective way to identify cancer genes. Very briefly, this works like this. If we have a cohort of tumors, for each of the genes, we just count how many mutations there are, and we also measure based on neutral mutagenesis, how the mutations occur and the mutation rates in those tumors, how many mutations we expect. And then we see if there are more mutations than expected. We also measure if there is a bias towards high functional impact mutations, or if there is a clustering of mutations in a linear or in a 3D structure of the protein. I'm explaining this in very simple terms. The most difficult part is always modeling correctly neutral mutagenesis and doing the statistics to see if there is more or different pattern than expected correctly. There is a lot of literature. I cite some of them here, but the concept is basically the one I explained. With these methods that can identify signals of positive selection in the pattern of mutations is the way that we generate the compendium of mutational cancer driver genes. Basically, we collect somatic mutations in many tumors, and this is public data from many different projects. And all this data, once curated and quality controlled, we run the pipeline, and then we identify cancer genes. At the moment, the current version has 633 cancer genes in 87 cancer types. Most interesting thing is that when you run these tools, this approach recovers most of the well-known cancer genes because they really have these very clear patterns. But because it's unbiased and it starts from the somatic mutations, it can also identify new genes or even identify genes that we knew that were involved maybe in breast cancer, and now we see the signals in 3 other cancer types. So all this information is publicly available in intogen.org. And it's not only a list of genes, it's actually a list of genes per cancer type, right? For these 80-something cancer types, you will have the list of genes that are available. So just feel free to go and navigate into Genome.org or download the data. We feel that we have quite a good list of cancer genes, especially for the cancer types that we have more data and are better studied. But we realize that when you go at the level of mutations, most mutations in cancer genes are of uncertain significance. Even mutations that we see in tumors, right? What we are seeing in this graph is mutations that we see in tumors in cancer genes. And we see that if we go to the databases, many of them are not annotated, and these are the ones that we usually call them variants of uncertain significance. And this poses a big problem when you are interpreting clinical information from a tumor sample. So we thought, in the same way, can we identify cancer driver mutations systematically from the data? And the solution here was we designed a machine learning approach inspired by evolutionary biology. This is called BoostDM. And it's basically thinking that all the tumor mutations in thousands of patients is like a natural experiment. Instead of doing experimental saturation mutagenesis, we think of this as a natural experiment that is occurring in the human population, and that we have a sample of that are the tumor mutations from the tumors that are available. And then we need to design it a way to learn which combination of features define driver mutations in each gene, each tumor type. I'm going to explain only the concept. Again, this is published, so anyone that is interested, you can ask later, or you can read the paper where there is all the technical information. But the most difficult thing here was to decide what is a good positive and negative set that is not unbiased, but is of high quality and that can define driver mutations and passenger mutations to learn, because at the end, this is a classification problem. So what we decided is to take the mutations observed in cancer genes in human tumors, because we can measure for each of them how much is the enrichment of driver mutations. This we do it with the DNDS approach. And we use all these mutations as proxy driver mutations, knowing that this is an imperfect positive set. To generate passenger mutations, it's very important to do it in a similar way as they occur in the tissue. So for every tissue, we simulate neutral mutagenesis to create a set of mutations as they occur before positive selection. And this is a set that is enriched for passenger mutations. Again, it's an imperfect data set, but then we design it, all the learning approach, all the machine learning approach, being aware that these are two imperfect data sets, but highly enriched in drivers and highly enriched in passengers. Okay, this is a very long story short, because I'm skipping a lot of details for the sake of time. But at the end, we have a model per gene tumor type. For example, what you see here is the model for EGFR in lung adenocarcinoma. Once we have this model, we can go to every single mutation, every possible mutation in the gene, and ask, due to the properties of this mutation, is this a likely driver or a passenger? And this way we can do what we call in silico saturation mutagenesis of cancer genes, that is annotating every single mutation, even mutations that we have never seen in tumors, as if they are drivers or passengers. And what you see below here are the some of the features that are used in the model. Again, this is publicly available in intogen.org/boostdm, and you can go and search the models and the blueprints for the different genes and tumor types. In addition of having an annotation and a score, a BoostDM score, we thought it was very important also that this is something that we can understand, and BoostDM explains each driver mutation through the combination of features. So for example, you can see here some examples that you see that they are called as driver because they are in a cluster of mutations in the 3D, because they are in a particular domain, they are conserved or not conserved, etc. In the paper, we did a lot of validation, comparing against different methods and different experimental saturation mutagenesis datasets to see that it performs very well and it outperforms the different methods. Again, many more details are in the paper. So what we show here is this is a result of one model, that is TP53 model, but this is the result of all the models that we have comparing BooSDM, and what you see in the y-axis is the area under the curve. So the higher it is, the better performance it has in identifying these mutations. Okay, so if we go back to the bars that I was showing before, I was showing you that in the databases we have a number of mutations annotated, but many that are not, the gray bars. If we now use BooSDM, we can see that we increase the number of annotated mutations. What you see in the left is 3,000 patients that we have whole genome or whole exome. In the right is the Project Genie, that they are panels. And that's the reason why the bars are a bit bigger, because they don't cover all the genes, and they cover the better well-annotated genes. You may notice this, there is a lot of gray bars still, so we are increasing the number of mutations that we can annotate, but we don't annotate them all. And the reason is that we haven't seen enough data of tumors to generate models for all cancer genes, right? There are some cancer genes that we don't have enough data and we cannot generate one BooSDM model. You see here also that I mentioned RankDM and RulesDMs. These are other tools that we also use to annotate when BooSDM are, for example, for indels and for others. But the thing is that there are some models and some genes that we cannot annotate, but the main reason is because we haven't seen enough data. So we are very confident that with enough data, these models can reach to all the genes. So what we are doing is we are collecting all these mutations in thousands of tumors. So for the moment, from public data sets, we generated this automatic learning platform, IntoGen, BooSDM, that generates this data, cancer genes and driver mutations. This is stored in databases. And this is what we use to annotate in Cancer Genome Interpreter. The idea of this new platform of Cancer Genome Interpreter is that it can help close this cycle, so that the data that is used, or at least part of the data that is used for the annotation of an individual patient, can also be part of the learning process, so that we can close these gray bars of unannotated mutations. So concept is more data, we generate more knowledge and better interpretation of mutations. So this project is a community-driven project. What we mean by that is that we have built it, you will see it from the presentation from Santi, together with different clinical centers in Europe. And for us, it's very important to balance the open science principles that I mentioned. Everything is published, is available in IntoGen and other databases, community-driven, and also we really aim for a clinical use of this platform. To achieve that, it's very important that the genomic interpretation is accessible, and making it accessible means, from one side, developing this CGI pipeline. We call this CGI pipeline the basic computational bioinformatics pipeline that enters a list of mutations, and using the databases from the automatic learning platform and expert knowledge, it annotates all these mutations with relevant knowledge. But it's not enough to have a pipeline, right? Because you will need bioinformaticians to run it, or you will need to install many things. So that's why we have developed the CGI platform that also contains a visual interface. We have worked on trying to make it the most intuitive as possible, so that you can understand what is there. But again, it's not enough, because this platform needs to be run somewhere and installed somewhere. And we would like this to make it very, very accessible for everyone. So that's why we have put it in a cloud, and it's ready to run in a cloud. And that's what we are presenting today, is the platform.cgi-clinics.eu. It's a ready platform. Just one note about the history of cancer genome interpreter. We started many years ago when there was no tools to solve this problem. And the project CGI-Clinics started in 2022. And we are in a bit more of the half of the project. So the project will run until 2028. So this week is an important milestone, is the release of this open platform. And this is something that we are still developing actively. So your input will be also important. I want to finish here and acknowledge a lot of people from the lab, but also the full CGI consortium community. And I'll stop here so that we can move to the next speaker.
Thank you, Nuria, for your presentation. So our next speaker would be Santiago Demajo, the scientific coordinator of CGI-Clinics, this major EU-funded initiative aimed at advancing precision cancer medicine through optimized genomic data interpretation. He is also a senior researcher at the Barcelona Biomedical Genomics Lab at the Institute for Research in Biomedicine, also known as IRB Barcelona. His research focuses on identifying the molecular drivers of cancer to translate genomic findings into actionable precision oncology. He has been instrumental in the development of key bioinformatics resources, including the Cancer Genome Interpreter tool. Dr. Demajo holds a PhD in biomedicine and a master's in bioinformatics and biostatistics. In addition to his research, he is a lecturer in translational cancer genomics at several universities. So, Santi, welcome, and we're looking forward to learn more about CGI from you.
Thank you a lot for the presentation. I will share my slides. I guess you see them correctly. So thank you everyone for joining. I will present now the new CGI platform and the CGI-Clinics project more in detail, as Nuria already highlighted some of these. So as Nuria was mentioning, to address the challenge of interpreting tumor mutations, we have developed the Cancer Genome Interpreter, a tool that was originally developed many years ago, but in the last 3 years or so, in the context of the CGI-Clinics project, we have revamped and we are now presenting the new CGI platform. So yes, I'm very happy to present today the new CGI platform with the objective of enhancing precision oncology to data-driven genomic interpretation. And before going into the platform, I would like to say a few words about the way that we follow to achieve this CGI platform, and this is in the context of the CGI-Clinics project. So CGI-Clinics is an EU-funded project of 5 years involving 5 countries, and it's a multidisciplinary network to improve precision oncology through enhanced genome interpretation. We are at the IRB Barcelona, we are in charge of the technical development of CGI, but we count also with experts in sequencing, patient associations, communication, and health economics and regulatory. And importantly, we closely collaborate with the 10 clinical partners that are part of the consortium. So one of the main characteristics of CGI-Clinics is this constant interplay with the clinical community. So we have developed the new tool following this constant interplay to be able to meet the needs in the clinics. So during the process, we have closely collaborated with the 10 hospitals of the consortium, and we are as well establishing more collaborations with other hospitals and initiatives. Entering now in the CGI platform, the main function of the platform, as already mentioned, is an advanced genomic interpretation system, so translating genomic data into actionable insights by identifying, on one hand, oncogenic mutations, and on the other hand, biomarkers of drug response. This is coupled with data-driven insights. So CGI includes inside the automatic learning platform that Nuria just presented, and the machine learning algorithms that are key to interpret variants of unknown significance. And this is complemented with a full suite of functionalities for genomic data interpretation, including reporting, virtual molecular tumor boards, and cohort-level statistics. To do this, we built this platform that is a secure, intuitive, and integrable professional-grade infrastructure with a new and intuitive interface designed to simplify the complex interpretation workflows, and that has been tested already with clinical users. It's a secure and GDPR-compliant infrastructure hosted in Amazon Cloud, meeting rigorous data protection standard. It works through private projects per each institution. So each institution may have one or more private workspaces, ensuring data isolation and privacy for each center. The platform includes a data and user management system. So you can manage both patient data and also project user permission and roles. And importantly, apart from the user interface, we have available an API for system integration that enables the connection with existing hospital or institutional systems and the automation of high throughput analysis. I will now describe a bit some of the main modules of the new CGI platform. These are the main modules: interpretation, virtual molecular tumor boards, statistics, my mutations, and admin, and I will focus in the main ones, starting with the core of the tool, that is the interpretation module. So what CGI does is to interpret the mutations identified in a tumor of a cancer patient, identifying the oncogenic mutations and the biomarkers of drug response linked to these mutations. To do this, the input of the tool is the mutations identified in the tumor in the form of a BCF file or similar, plus some basic clinical data such as the tumor type. And for the interpretation analysis, CGI uses a combination of expert knowledge in the form of databases and the automatic learning platform, that is the compendium of algorithms such as Intogen and BoostDM that Nuria introduced. So this would be the results of interpreting the mutations from a tumor. This is an example of a lung tumor. Here at the left, you see some basic IDs and some basic clinical data associated to this tumor. And in the results part, at the top part, you see these relevant alterations, which are the main mutations identified are relevant by the system, by CGI. In this case, is these 4 mutations. And in this part, you see the 2 main blocks of results: the alterations, so the mutation classification, and the biomarkers of drug response. So focusing a bit more in the alterations, in this case, you only see small mutations, but if the tumor has copy number and fusions, this will also be displayed as different tabs here. So what you see here is all the list of mutations identified in the tumor, annotated and classified as oncogenic and non-oncogenic. For this classification, the tool uses, on one hand, expert knowledge in the form of different databases. For example, we have some oncogenicity databases such as ClinVar, CGI, OncoKB, and some gene-specific databases included in the tool. Recurrence databases such as CBioPortal, Genie, Cancer Hotspots, to see whether the mutation was observed previously in cancer patients, and other databases like population databases such as NOMAD and clonal hematopoiesis. And this is combined with the automatic learning platform, Intogen to annotate cancer driver genes, and BoostDM, RankDM, and RulesDM to annotate cancer driver mutations. And the 2 elements are combined in this final classification of the mutation. The second block of the results is the identification of biomarkers of drug response with different levels of evidence following, on one hand, AMP-ASCO-CAP framework and also SCAT framework. So what you see here is the list of biomarkers identified in this tumor. In this case, there are quite a lot because we have these very typical mutations in EGFR. So for each biomarker, you have the mutation, the drug, whether the mutation leads to responsiveness or resistance to the drug, the evidence level following the AMP-ASCO-CAP system, and the SCAT scale for clinical actionability. And the source of evidence here is these three. We have an internal CGI biomarkers database and also CIVIC and OncoKB. So you can also explore more in detail the interpretation results and focus in each of the mutations. We have here one of the mutations identified, this very typical mutation in lung cancer in EGFR. And what you see here is the classification of the mutation and specifically the classification and recurrence in expert databases such as ClinVar, OncoKB, CBioPortal, Gini, etc. And on the other hand, the automatic learning platform annotation. In this case, this mutation is annotated as driver by BoostDM with a very high score, and also the biomarkers of drug response linked to this mutation. After interpreting after getting the interpretation results, CGI also allows to generate editable reports based in ESMO recommendations. This is how a report would look like, with the patient and sample details in this part, followed by the oncogenic mutations identified, in this case, these 4 mutations, the clinical actionability, so the list of biomarkers, and other relevant information. Going now briefly into 2 more important new modules. One of them is the virtual molecular tumor board. This will be explained a bit more also by Loic, but the idea is that this module facilitates discussion of complex cases among experts. It's managed by the CGI virtual MTB managers led by the VHAO in Barcelona. We count with a database of international experts of about 50 experts at the moment. The tool includes a teleconference system integrated and a slide format report to present the case. And currently, the CGI virtual MTBs have these 2 parts. One that is what we call clinical part, based in an IVD or ISO compliant report. And the second that is an educational part, discussing the CGI report that currently is not yet clinically approved. And finally, some words about another important new module, this is the statistics module. This module allows to explore and analyze all the data that you have introduced in your project. So you can explore all your patient, sample, and mutational data, apply different filterings, and visualize and download all cohort level data and produce some plots on the data. So now I will share a short demo showing how the full workflow was to analyze mutation from a patient tumor would work in CGI.
Welcome to the CGI platform. This is the homepage of the platform, in which we have direct access to some of the main functionalities, the last analysis in our projects, and other features. To interpret the mutations identified in a tumor, we should go to the Analysis module, click on New Analysis, and enter some basic information required for the analysis, such as the IDs, the type of sequencing used in the analysis, and the tumor type of the sample. Then we need to upload the file with the mutations. We can drag the file or select it from our folders, and then we run the analysis. In this screen, the results of the analysis will be displayed. Now let's take a look at the results of another analysis. This is the results page. In the left column, we have the IDs and the most relevant clinical data. In the upper part of the screen, we have the relevant alterations. These are the alterations that have been identified as oncogenic. We can expand the information of these mutations, and we can show the algorithms and databases used for the classification and the annotation. In this table, we have the complete list of the mutations analyzed. All of them have been classified and annotated by several databases and prediction algorithms, and here we have all the related information. In this table at the bottom, we have the biomarkers of drug response linked with the mutations identified in the patient. After reviewing the results, we can generate a PDF report that contains the basic patient information, the identified oncogenic mutations, and biomarkers of drug response, as well as some other sections that can be personalized. Additionally, we can request a virtual molecular tumour board that consists of a meeting with one or several experts to discuss this specific case. Here we enter some information, we select the availability slots, and we send a request. In the Patients module, we have the complete list of the cohort of patients analysed in the platform. For example, for this patient, we have the complete clinical information and the associated analysis. In the Statistics module, we can display the complete cohort of patients and all the mutations analysed in our projects. In My Mutations, we can analyse single mutations, and in the Admin module, we can manage the projects in which we are administrators.
Okay. After this short demo, I will focus the last part of my presentation in some ongoing and future plans for the CGI platform, the CGI-Clinics project. One important part ongoing in the project is the performance evaluation and clinical validation of the new CGI platform. I will not enter in details in the results because this is still ongoing, but I will share with you the different types of analysis that we are doing. So on one hand, we are comparing CGI classification with reference databases such as ClinVar and OncoKB, using public cohort data like Gini and comparing the classification by CGI and the databases. On the other hand, we're evaluating CGI performance in real-world clinical settings, so comparing CGI classification versus hospital interpretation procedures using each different hospital cohorts. And finally, another group of analysis that we are doing is trying to clinically validate VUSs that are reclassified to oncogenic by CGI by studying different cohorts and studying patients with known oncogenic mutations compared with patients with VUSs that CGI reclassified as oncogenic, and evaluating its potential clinical potential. So preliminary results look very good, and we hope to share them very soon. Another important aspect we want to share is that we are in the 4th year of the project, but the CGI platform will continue evolving together with the precision oncology community, and we expect to have an even better version by the end of the project at the end of 2027, and we would like to do this by continuing interacting with the precision oncology community to keep improving the tool in the future. We are still designing this new iteration of the tool, but one thing that we already are starting to implement is the connection with clinical trials. So we will include, we will link CGI with some clinical trial matcher tools so that when you interpret the mutations from a tumor, you will be able to see clinical trials in which this patient or this tumor could fit. This is something we are already working in. Another important milestone of the project that we are sharing today is that we have just released what we call Open CGI. So this open version of the CGI platform is thought to be used by the cancer genomics research community, and our objective is to extend the use of CGI to the whole community. To, on one hand, contribute to cancer genomics research, and on the other hand, keep on learning from the community and improving the tool in the future. So we have this CGI Open is a freely accessible version with autonomous user registration for immediate use. So basically, you can enter the CGI-Clinics website, access the CGI platform, register, and start using it. We upgraded the cloud infrastructure to ensure the stability for extended number of users to be able to use it. And as I was mentioning, we're open to feedback to keep improving the tool. This is the difference between what we call CGI Open and CGI Teams. That is basically the tool that I explained up to now. So in the CGI Open, you will have a limited analysis and storage capacity compared with the CGI Teams. The CGI Open is thought for single user project. Instead, the CGI Teams can accommodate collaborative projects of up to 30 users. And in CGI Open, some features are unavailable, such as the virtual MTB and the reporting. And finally, another important future piece we want to put here is that we are following the regulatory pathway to achieve IVDR certification for the CGI as a medical device software. And in this point, it's important to highlight this disclaimer. So currently, the CGI tool does not have CE marking, not any other medical regulatory authorization. And this is why the CGI tool is provided strictly for research use only. But we are transitioning to IVDR certification as a medical device software, as I was commenting. So now we are here. We have this CGI Teams version that is not certified for clinical diagnostic use. That is the version that is available and running in our 10 clinical partners plus other collaborators. But we expect by 2028 to have a CE-marked medical device software intended for clinical diagnostics. And just to finish, how you can access the CGI platform. There are these two ways. For academic research, we have the CGI Open. To access the CGI Open, you just need to go to platform.cgiclinics.eu, register and accept terms and conditions, and you can start immediately using it. And if you are in a clinical setting or you want a collaborative project, you can request access to CGI Teams. To do this, you would need to fill out this form. And we will give you access to the CGI Teams with a 3-month demo version. And in the meantime, you will need to sign a free license and data processing agreement. And note that this CGI Teams is the version that is GDPR compliant for private patient data, differently from the CGI Open. And importantly, we are, as Nuria will also mention at the end of the presentation or of the webinar, we are expanding in the last phase of the CGI-Clinics project. We are expanding the CGI-Clinics network to up to 30 hospitals. So if you are interested, please get in touch. And to finish, just some take-home messages. We developed the new CGI platform through a collaborative effort within the CGI-Clinics project, and we will keep improving it. The CGI platform includes advanced genomic interpretation together with functionalities such as virtual MTBs and cohort statistics. We are following the regulatory pathway to certify CGI as a medical device. And you can access the CGI platform by either registering to the newly released CGI Open or by requesting access to the CGI Teams. And just to finish, I would like to thank the people from the Barcelona Biomedical Genomics Lab at IRB Barcelona and all the CGI-Clinics consortium, and patients and families, of course, and funding agencies. Thank you a lot for your attention.
Thank you, Santi, for your presentation. Next is Loic Verlingue, a physician scientist involved in the field of medical oncology, clinical research, and digital healthcare. He is an investigator in early phase clinical trials at the phase 1 unit and in the precision oncology group of Centre Léon Bérard in Lyon. He is head of the data science group in casting team at the Centre de Research and Cancerology de Lyon, or CRCL. His topic of research are natural language processing on medical text and artificial intelligence on molecular data for precision oncology. Loic, we're looking forward to hearing your presentation.
Many thanks, Alexandra, for the nice introduction and the invitation from the panel. What I want to share with you today is a practical view of how we could use CGI in an hospital. And of course, I will speak of our experience at Centre Léon Bérard. So my outline is first introduce you to the Molecular Tumour Board at Centre Léon Bérard and the genomic data we are producing. Then give you a glimpse of our experience, initial experience with CGI Virtual Molecular Tumour Board. Then a bit of a perspective in terms of benchmark of variant interpretation, what has been done so far and what should be done, I think. And finally, a bit other perspective on how we could connect CGI to and how it can help to do some other research and connecting to other tools, mostly AI tools, as Alexandra stated from the introduction. So molecular tumor boards, it's very basic. Sorry for the repetition. I'm sure everybody of you knows, but molecular tumor boards has changed from the last years. And it's always handling molecular data of patients affected by tumors, by cancers, in order to orient them or to propose molecular targeted agents as standard treatments, as early access, but most often it's to orient patients to clinical trials. There's a lot also about prevention, because we often found out some potential germline variants thanks to those somatic analysis. And in fact, we are more and more dealing with different types of data. Of course, always the clinical reports, imaging, plus the molecular data that have expanded through the years, from TargetSeq to now whole genome, and new techniques that I will briefly touch. And we also deal with technology allowing us to do trial matching, to discuss with international experts, like in the virtual molecular tumor boards. And CGI is proposing a tool that is efficient, I will try to show. And there are several other European networks, European projects that are building some initiative of virtual molecular tumor boards using tools such as CGI. So I think it will last for the next year and have a broad impact in our community. So at Centre Léon Bérard, in my hospital, we have quite various type of molecular analysis. So one of the, I would say, classical program is called Profiler here. It has run for 13 years right now. And it analyzes DNA, RNA for patients affected by refractory or metastatic cancer, and we analyze around 700 patients a year, and it's regional. So we centralize the analysis of the patient in the region, and now even in other region. Here at CLB, and we centralize the molecular tumor board. We also have national initiatives such as the France Medicine Genomic Program 2025. We have, of course, ctDNA sequencing. So with interesting technologies that have been developed in-house with targeted seq, of course, but also analysis of copy number variation and fusion analysis from circulating tumor DNA. And we also have some, of course, routine analysis and research analysis with new techniques that I've just mentioned, like single-cell RNA-seq, digital pathology, and potentially in the future spatial transcriptomics that, of course, are very present in the research, but here I present the data that we then analyze in the context of a molecular tumor board, either as a translational research, but also for, of course, treatment decision for the patient. So what is the impact and why MTBs are important? I'm really convinced that MTBs are here to promote local, regional access to clinical trials, and there are also a big topic about cross-border clinical trials. So here is a map of France, and we are here in Lyon, and we are very close to borders with Switzerland, Italy, not far from Spain, not far from Germany, etc. So this is the area of recruitment of our patient in our clinical trials as CLB. So and this is molecular analysis are very important to orient patients to clinical trials. So we have to also work on the frontiers, on the borders, to allow patients to access to clinical trials. So it's from very local to national and now international, and for the international part, virtual molecular tumor boards and European projects are very important. And we have a dedicated project, EACR, for improving cross-border access to clinical trials. Our bioinformatic pipeline here, led by Anthony Ferrari, who is lead of the bioinformatic platform, is, I think, will ring a bell for most of you. So this is a profiler. So this is in the profiler program, the classical program we have. So we analyze mutation, copy number variation, fusion, and expression from RNA-seq. And there is a step of annotation by molecular biologists, and classically they use Alamut, and now more and more MOBI details that allow access to databases and annotation tools. And of course, we are here to discuss about CGI. And of course, so we have been implementing for a few months now CGI in this pipeline. So it's Camille Brodin who is developing the connection and the implementation. And so she uses the API developed by the CGI developers to run the analysis on mutation data, CNVs, and fusion. And this is done in parallel of the annotation by molecular biologists. So this is, as we have to stress, this is a research part, and the main usage of this result for now is to compare and to evaluate the performance of CGI compared to the classical tool our biologists are using. And of course, at the end, it goes to Molecular Tumour Boards. So thanks to this organization we have had for some months, we have been able, and Camille as first initiator has been able to provide some presentations, some posters about the evaluation of CGI. So this first poster, we did a combined analysis with Gustave Roussy Cancer Center in Paris, in Villejuif. And the second one was at ESMO AI, so that would be the end of my presentation about how we can connect CGI to other AI tools to make a multi-agentic workflow for our future potential molecular tumor boards. So virtual molecular tumor board, I think, is a great tool for now. It's a bit a primer in the EU. There have been few literature in other countries for molecular tumor boards, international molecular tumor boards, and the potential impact are, of course, quite obvious. The increase on the expertise on difficult case specifically. So for example, difficult molecular interpretations or rare clinical situations of rare tumors. I will show one just after. But also, as I just presented before, increase the option of clinical trials for patients in the EU. And of course, it's also the way and the place to discuss the tools so CGI, their usage connected with the hospital, with the physicians, with the molecular biologists. And this is a good brainstorming and evaluation time and place to discuss the tool. So as Santi just presented to you, the virtual molecular tumor board is in two parts. So there is a first discussion on the CE marked, ISO compliant, etc. Analysis. So as with the analysis we do locally, and there is an educational part to evaluate the output of CGI in the context of the patient case. So CGI, vMTB, you can request it directly from CGI tool based on an analysis you run, and there is a way to book a time slot with the experts that are available at this time to discuss the cases, and we are around 4 to 6 or even more sometimes to discuss 1 or 2 cases when it's needed. So this is one of the case we've discussed recently. So the question I asked to the panel, so vMTB panel, was: are there some clinical trials option for these patients? You will see why, because the patient is a patient affected by chondrosarcoma, mainly undifferentiated one, and known IDH1 mutated that was found here in the analysis. So it's R132C classical hotspot mutation in IDH1. And the patient has been refractory to 3 or 4 lines of therapies, primary refractories. So the question and the problem we had is there are still no access to IDH inhibitor like ifosidenib in the approved setting, and no reimbursement for now. So that's why we ask for clinical trials, and we have locally and in several sites in the EU a clinical trial called Conquer, but it's restricted to first line treatment patients, which was not the case of this patient. And from the publication, we know that treatment with ivosidenib or IDH1 inhibitor for this type of hard to treat tumor lead to 26 overall response rate. So this is a very effective therapy that we would like to offer to our patients. So the one conclusion of the vMTB was try to ask compassionate use from Servier, the company who is developing and handling ivosidenib. And there were also another interesting fact that there were 11 mutation per megabase, which doesn't seems very high, but for chondrosarcoma could be a bit interesting in terms of high mutational burden or quite high, I would say. And there was also this immunosarc trial that have been published, and the trial is finished now, combining sunitinib with nivo, leading to good overall response rate too. So that was an option, but also out of the recommendation for now. And there are clinical trials that we had at the time at CLB, like MQ01, oriented on TME. So that could help to brainstorm about the potential treatment option for this patient. Then I'll move to the benchmark. So does CGI work still the best, as Nuria showed? And for that, so the field is advancing very fast. So Nuria showed that CGI was in the very first, or at least Intogen was in the very first tool at the very beginning of the history of variant interpreters for cancer, at least. But the field is advancing very fast, mostly with AI tools, for example, with AlphaFold, so you see AlphaMinSense, and there are also LLM-based models that have learned to infer some variant interpretation, like EVO2 or ESM1 and 2. So this is another figure that from the papers that Nuria was referring to in Nature. That's for PIK3CA, showing that BoostDM AUC score in terms of performance is very high for different tumor types. And what I like is that there is this ESM1B tool that was evaluated, which is the first generation of LLMs for such tasks, but still not working so well by the time on such PIK3CA variant annotation. But this I think needs to be updated given the new tools. And finally, CGI-Clinics has, I think, very high potential to be connected to other tools. So Santi mentioned about clinical trial matching, and one of the initiatives we have currently is combining it to a clinical trial matching tool and company called Clineo. And one other very advantageous situation is that CGI is currently connected to 10 hospitals and will expand by the end of the project to 30 hospitals, which means that you have the data of 30 molecular data of 30 hospitals connected to a cloud service, which is highly secured, etc., GDPR compliant, etc. But it means that it's a data lake, if I could say, that can potentially allow other tools, and we have different tools that we are developing in my team, like PubMed request, digital pathology, CT scan, etc. And that's how we've envisioned the future use, complete use of CGI plus other data to make good recommendation in terms of treatment and clinical trials. So that's a vision I'm trying to emphasize in the EU, because we have many challenges in terms of access to clinical trials and also country readiness to clinical trials, so that the companies can come and come more often to us in the EU to propose new treatments for the development, at least in phase 1, because that's where I'm working. And some initiatives that could help a lot is this data generation and sharing connected to trial matching tools. So informing the patient and the physician about the trials that their patient could be referred to, and efficient infrastructure in terms of clinical trials, and that there are several EU projects that are working on that to improve the infrastructure of personalized cancer trials like Primrose, GAPCM, Spiderweb. So with that, I would like to thank the many people I'm working with locally, specifically Professor Pierre Santini, who is leading this huge team we are in, the Molecular Tumour Board at CLB with the faces here, the founders, CGI team for their confidence, and thank you for your attention.
Thank you, Loic, for your presentation. I'd like to invite Nuria now back, as she'd like to say a few more words to bring all the presentations together and nicely wrap up the project introduction.
Yeah, I have only 3 more slides, but it's basically to inform that Loic already mentioned it, that we are extending now CGI-Clinics network to additional number of hospitals. And basically we have an open call, and candidate hospitals that are performing NGS have some legal support to just address all the issues, and have a CGI specialist, basically someone that can make the bridge between our teams and the hospital, are allowed now to join the network. And basically, if you're interested, just go to the web, cgiclinics.eu/cancer genome interpreter, and you will find a screenshot like this. And if you want to join CGI Open, just go register and you can use it 1 minute later. If you want to join CGI Teams, you just fill up a form, this request CGI Teams, and we will give you access. And one more call that we are opening, and this is a very important one. It's going to be open in 15 April. And this is because as part of the project, we also have funding for sequencing. And we are creating this CGI 500 Genomes Initiative, which is a collaborative initiative that aims to build this data set with 500 whole genomes, whole exomes from tumors with scientific or clinical interest. And we are asking the community that are interested in participating, have some interesting samples that would like to sequence, to participate in the call. The call is, the different projects are going to be evaluated by a panel of experts, and the selected ones are going to have the sequencing, the full process included. So you're going to send a sample to the sequencing center in Barcelona, the CENAC, and this is going to be analyzed. So it's going to be sequenced, analyzed, interpreted, and you will have the results in Cancer Genome Interpreter. And we are also requesting that this data is ready to be shared for the broader community. We're going to put the data in EGA. This is a key element for this call, is also aiming for data sharing broadly. So the samples that are proposed for this initiative need to have ethical approval for control access for a repository. So just be aware of the two things. Open call for anyone that would like to use the CGI teams, just fill up the form. And soon we'll open this call with a bit more details, but if you are interested, just be attend to the call. And that's all from our side. So we are ready for questions if there are.
Thank you, Nuria, and to all our speakers for your excellent talks. I can see they have generated quite a few questions for discussion already. I'm going to get straight into the questions asked, but please do add the name of the speakers to your question if you want to address the question to someone specifically. We aim to take a question from for all the talks pretty much. And as mentioned earlier, we would love for this webinar to be interactive, so we invite all of you to participate. I'll briefly give you the information on how to access the Q&A box just in case you joined us a bit later. So you'll notice there's a Q&A button at the bottom of your screen. If you can't find it, it might be hiding under the More button represented by the 3 dots. If you click on that button, a Q&A box will pop up. You can add questions at any time during the panel discussion, so we already have a few questions, but you can also type them in now or as we answer some of them. You can also ask questions anonymously, and I will read them out loud, but once again, we would love for this conversation to be interactive. Questions that are left unanswered, we'll try to get back to you with the answers as much as we can. So I'll invite our speakers if they want to have a look at the Q&A box, because I think some of the questions might have been answered through the presentations already. So would you be happy to start with Diana's question, or has that been answered?
Yeah, Diana's question is, can the drivers be seen?
I can add Diana to the conversation if she's available. Diana, would you like to open your mic?
I was interested in the translational aspect. I mean, I speak as a patient carer, not as an academic. Can the drivers that Nuria referred to be seen? In other words, are they reflected in MRI scans to aid diagnosis?
Yeah, Diana, no. So the mutations and the drivers need to require NGS, so next generation sequencing, DNA from the tumors and sequencing, and this is the way to this is the input data for our CGI.
Thank you, Diana, for your question. The next on my list is Andre Duarte. I'm hoping I'm pronouncing your name right, Andre. And I can see you've got quite a few questions, like 3, I think I've counted, so feel free to ask all of them. added you to the panel, so if you can access your microphone, please go ahead. I'll start with the first question. So what is the data policy for using the CGI platform?
Maybe Olivia, you can
Yes.
answer this.
I can take that one. Probably he's referring on how he can be using the platform and how IRB or the CGI team is using the data by requesting your CGI demo. Your data is not used, then you can directly log in and register login and use the platform directly. This is the first step we recommend to everyone that wants to have a first glance of the platform. Then if you like it, please go to CGI Teams form in the website of CGI-Clinics and request for a Teams version. And with this, we will provide a license of CGI and a data processing agreement where the conditions from our side, how we process the data, and the conditions that we request from your side, for instance, pseudonymization of the data, are settled and established in this documentation.
Thanks, Olivia. Andre, if you want to join the conversation, feel free at any time. Another question that Andre had was, how does the CGI platform handle mutations with low VAF?
Santi?
I think I could take this one. Yeah, for the moment, one thing that we need to highlight is that CGI is what is called tertiary analysis tool. So CGI expects already the primary and secondary analysis to be performed, and the input of CGI should be the somatic mutations identified in the tumor. Already excluding, for example, artifacts with very low BAF, etc. Regarding real mutations with low BAF, for the moment, we are interpreting them as the rest. So a new feature that we have in the tool now is that we display the BAF in the results. So user, when doing the interpretation, can check the BAF of that mutation. But for the moment, we are not filtering out mutations with low BAF, and it's the user that needs to do this. What we do have is like a column in the interpretation table that in which the user can highlight, can annotate some specific features of the mutations, and one of them is marking the mutations as low BAF. And regarding future versions, we are working in including more about BAF filtering and taking BAF more into account in the interpretation.
And can the CGI platform identify hypermutated tumors?
For the moment, we don't have any specific. For example, in other tools like Intogen and BoostDM, we do have some filters for hypermutated tumors to take this into account, to include or exclude them for our learning pipelines. But in CGI, we do not have any specific feature related with hypermutated tumors.
Thank you. The next few questions are from Guillermo. Guillermo, if you're able to join us for the conversation.
Yes, hello.
Hi. So would you like to go ahead and ask your questions now?
Sure. So, well, first, thank you for the presentation. I have some brief questions. So the first one is regarding the non-coding part of the genome. If you are interested in enhancing your models like BoostDM for these non-coding mutations.
Yeah, very quick answer here. It's very difficult. We are interested, but it's a very difficult topic. At the moment, it annotates very clear, like TER promoter mutations are annotated manually, but BoostDM cannot deal with non-coding mutations at the moment. Okay, we would like, but it's a very difficult research question.
Okay, thank you. My second question is related to when you have the report of the different drugs available for that specific mutation. If there are multiple drugs for one mutation for a specific patient, is there a way to rank them or prioritize them or this is a oncologist type of thing that he or she has to do?
This is a good point and very interesting. We have some functionalities, but we don't have a rank per se. So, for example, when there can be cases of drugs that are repeated from different sources or biomarkers that are repeated. So this we have a functionality about this, and also we have a functionality of contradictory biomarkers, because the same patient, and one of the examples is actually the example I showed, may have one mutation leading to responsiveness to a drug and another mutation leading to resistance to the same drug. So this is highlighted in the results. But for the other things is more on the oncologist side to decide. But it's a good point, and we are actually working in some kind of prioritization or best ways of displaying the drugs, that in some cases for very typical mutations there can be a lot of different drugs available.
Okay, so you don't have a prioritization, but you already have some scores for drug response, no?
Yeah.
Okay, thanks. So my third question is related. I think you said something, Santi, about this, but in terms of how the sequencing quality impacts the results. No, if the sample has low purity, for instance, or the bioinformatic team didn't properly, remove specific low quality variants and so on. I guess that CGI doesn't do anything. Just to ask.
Yeah, this is another very, very important point related to the VAF that I mentioned before. This is something that we are working in, but for the moment, we have some very basic controls, more about formats and to remove things that are very clearly wrong. But we are working in improving this filter. So the idea is that this quality control analysis should be done before the mutation is included in CGI for the moment. Like this could be part of this secondary analysis, and this should be done previously. But even though, considering this, we are working in adding some functionalities in the future version regarding quality controls, at least to display some warnings if there are strange things in the sample or things in this line.
Okay, just one quick note on that as well. This is for interpretation, but for data that is used in automatic learning platform, then we have very strict quality controls. So many of the data that is used for interpretation may not be used for learning because we always need tumor and normal and high quality data. So we have some quality filters to get into the learning process.
Okay, makes sense. Thank you. And my last question is regarding if you plan to expand like other types of analysis, like mutational signatures, or this is still very early on for research purposes only.
Santi, you want to tackle or
I mean, we have a lot of plans. We always have a lot of plans for the future. Mutational signatures are in our list. We probably will not tackle this very short term, but other things that we are working in, for example, is addressing tumor TMB or MSI. That I saw this also in one of the questions. So these things that have a really clear clinical implication, we are going to prioritize to address for the near future versions. Not that much in the signatures, although data is very promising, but it's not that well established the technical use. So we are prioritizing more the things that are more clinically relevant, let's say.
Okay, thank you so much.
Thank you, Guillermo. I can see that some of the questions that Xavier asked resonated with a few people from the audience. So, Xavier, if you're available to talk, would you mind asking your questions live?
Yeah, hello, can you hear me?
Yes, we can hear you. Thank you for joining us.
Thank you. So first of all, I'd like to thank Nuria and Santi for the presentation, for the amazing tool they have developed, and we're looking forward to using it very soon. So I had a question regarding because you said when you upload a list of mutations for a given patient, you specify the tumor type. So what happens, and that I guess it helps the tool to sort of try to identify those that are oncogenic and those that are not for that tumor type. But what happens when we have a tumor of unknown origin? Is there any specific approach that the tool uses for those type of tumors, which are kind of frustrating because you don't really know what you're dealing with? Or, I mean, how does the tool handle those type of tumors?
Santi, you go.
Yes. I mean, we don't have anything very specific for this. What we have is a lot of functionalities that are, I mean, there are a lot of functionalities that are tumor type specific. And as you said, by selecting the tumor type of your sample, you will get a much accurate interpretation. But we also have all the functionalities have, let's say, a backup. So if you don't know the tumor type of the sample, or if it's a tumor of unknown primary, we have, for example, models, BooSDM models that we call general models, that we call that are not tumor type specific, but cancer general models. And these ones could be the ones used for those cases for where the tumor type is not known.
Okay, I guess one thing that would be really interesting is if the tool could try to guide you based on the mutational profile of the sample that you have uploaded. If it's a tumor of unknown origin, try to guide you based on the mutational profile to the potential tumor type that sample is coming from, so this looks like a colorectal tumor, or this looks like a pancreatic tumor. That would be really helpful, because when at the Molecular Tumour Board we deal with these tumors of unknown origin, it's like, okay, yeah, the pathologist says it's a gastric tumor, but you never know, right? So maybe having the molecular profile talk a little bit about the origin would be really helpful. So another thing was about the SCAT classification of alterations, which is sort of difficult to work with, and it's also very dynamic, based on the currently data available at any given time. So how do you plan on keeping the SCAT classification updated in your platform?
Yeah, I mean, this is a very important point, not only for SCAT, but in general, all knowledge and all databases are very dynamic. For the biomarkers part, we are, for the moment, we are a bit in the pre-AI era, let's say, and we are still doing like manual curation of drug approvals, etc. And regarding SCAT, the same. We are doing this. We have people involved in the curation of the guidelines and including SCAT. So this is we have this internal CGI biomarkers database that we keep improving and updating quite frequently. So we will be adding new SCAT levels when the guidelines are updated, etc.
Okay,
and for just one quick comment, for the annotation of biomarkers, and this is manually curated at the moment. We have a parallel project to use agents, AI agents, to annotate this automatically. So this is starting, but will come at some more.
Yeah. Great. And then the last question. So Guillem asked before about, you mentioned before TMB and MSI as complex markers that the tool will be able to generate. But what about HRD, which is more used every day? And will the tool be able to generate HRD scores for those panels that allow it?
Yeah, I mean, it is also in our to do this. We will try. As you know, there are some limitations regarding whether the input data comes from whole exome, whole genome, or in some cases there are, as you said, panels that allow these calculations. Yeah, this is in our plans to add these new biomarkers that, as you said, are very used in the clinics, and hopefully we can include this in the next version of the tool.
Okay, great, and thanks again.
Thank you, Xavier, and thank you everyone for your questions and contribution. Do you think we've got time for one more question, or maybe not?
Fine for us, at least for me.
Okay, so another question that seemed to resonate with a few other participants came from Michael Dung. So Michael, if you're available to ask your question.
Yeah, can you hear me?
Yeah, we can hear you. Hello.
First of all, thank you for your work. Yeah, my question is, you mentioned that the data has to first be worked over by bioinformaticians after sequencing, if I understood correctly. I saw on your platform that we then upload the patient genomic data. Just on what format does this file have to work on your platform for analysis?
Yeah, the format is.
I'll let go Santi, and then I add something at the end.
Yeah, so the basic format is at the level of BCF. So it can be a BCF format or similar. It can also be a TSV, but the idea is that so the input should be the list of mutations, of somatic mutations in the tumor. One important thing for more that is ongoing and will be part of CGI in the future is the connection also with pipelines that do the full analysis. So we are connecting, for example, with maybe the Hardwick Medical Foundation pipeline that they are experts in analyzing whole genome sequencing. So we are already working in connecting our interpretation tool to some pipelines that do the analysis from the raw data, from the mutational raw data, to be able to cover the full spectrum. And basically, this is what we will apply in this 500 CGI 500 initiative, covering from the FASTQ to the interpretation result. But as I was saying, currently the file that you need to enter is at the level of BCF.
Nothing to add.
Thank you.
Thank you very much, Michael. Another question came from, I apologize in advance, Pornpimo. I may not have pronounced that right. Are you available to use your microphone?
Did you hear me?
Yes, we can hear you.
Okay, thank you so much. Thank you so much for a really nice presentation and demonstration. And also thank you that I have a chance to work with the CGI team demo version a bit. I have a few questions here. For example, in the clinical trial and the clinical routine use, we have, for example, we have longitudinal data from the same patient with the different treatment or the different time point. This is possible that in the CGI platform also show how we can tracking the identification of mutation that is persistent over time or the loss over time. Yeah.
Yes. So currently, the platform is structured to be able to organize all this data in the patients. So in each patient, you can create different samples. For example, if they are longitudinal samples, so samples included in different time points, and perform interpretation analysis in different time points. the interpretation analysis per se are independent, so it's they are not talking to each other for the moment, but at least you will have all the analysis together in the merged, let's say, in the same patient.
Okay. Right now, let's say right now the data not connect to each other, no? Time point 1 not connect to time point 2, but all of them the same sample, same patient, let's say have a 2 measurement. Okay, because this way, for example, I see the advantage. For example, I can track for the chronology, right? Which the mutation still occur over the time.
Exactly.
And then another part is also related to the clinical trial. For example, I saw you have a function called my mutation. No, in the my mutation, I can put let's say the specific biomarker that I'm interested, and I see has also the let's say function called the classification. If I classify this mutation for our clinical trial, and I presume this is a driver mutation, if I put this information in there, when I have a new patient from the same clinical trial, this information will be included for the interpretation, right?
This is a very good point. I didn't have time to enter in this, but yes, we have this kind of functionality. It's not through the My Mutation module, but through another functionality that is called internal databases. So when you configure your CGI project, you will see that there will be a tab of internal databases, and you can include a database of mutations that you have interpreted, so that when you, for example, is let's say a list of 1,000 mutations annotated by you. So when you see one of these mutations in your interpretation of your next patient, you will see also a column in the results related to your own internal interpretation. So this is this is a functionality.
Thank you so much. This is really cool. I'm looking forward to try. Thank you.
Thank you.
Thank you for your questions. Next would be Jordi. Just give me 1 second, and Jordi, can you use your microphone now?
Hello.
Hi.
Hi. Thanks for the presentation. I have a question regarding the low frequency variants. How the CGI manage low frequency variants? You can do some clusterings in order to find heterogeneous heterogeneous clusters of the tumor, or something special in this context.
Do you mean low variant allele frequency or
Exactly.
rare mutations across a cohort?
Low variant allele frequency.
Okay. Maybe Santi, you can.
Yeah, I think this is similar to what we discussed before. So for the moment, the only thing CGI does is to display the VAF at the results. So it's the user that needs to filter or interpret the VAF according to their knowledge. And in the future, we are planning to include some filters or warnings when the VAF is very low, for example, below 5%, etc.
Thank you. And the next question is regarding if I have, I don't know, in my sample, 5 variants related with cancer, and I would like to know the probability to this sample could be cancer related, for example, having some additive score or something in this context. Do you have some analysis that can do internally in CGI for additive score or combination of many genetic variants?
So I can take it. So the tool assumes that that the sample is a cancer sample. So we are not predicting whether it's a tumor sample. We expect all samples to be tumor samples. Yeah.
Okay. No, I mentioned this because, for example, with cell therapy products, sometimes you don't know if the sample could be tumorigenical or not, the product itself. And I was thinking about if the two could say something or have some indication in this context? Just for this comes my question. So we have to assume that the sample that we introduce into the system is tumorigenic.
Yes, exactly.
Yeah. Mhm.
Okay, thank you.
Thank you very much as well. I'm aware that we're quite a bit over the time, and I'm grateful to everyone for the questions, and I'm so glad that the webinar started such a lively conversation. I'm also mindful of our invited speakers' time. So thank you everyone for the lovely conversation. If your question has not been answered, I encourage you to ask your questions and any other questions you might have and address them to the CGI team through the contact form on their website. The link is in the chat if you're interested. I'll take this opportunity to remind everyone that the EACR organizes an annual congress for cancer researchers. The EACR 2026 Congress, Innovative Cancer Science, will take place in Budapest on the 8th to the 11th of June 2026, and we'll have a satellite event on the 7th and 8th of June, which is dedicated to early career researchers. The early rate registration closes on the 21st of April, so there's still a bit of time to register if you're interested in joining. You may also be interested in our Cancer Genomics, Multiomics, and Computational Biology conference, organized under Nuria's supervision. Registration is still open for a few days and will close on the 13th of April, should you be interested to join us for that conference. Thanks again, everyone, for your questions and our panelists for the presentations. Is there anything else that any of you would like to add?
No, only thanks everyone for attending and for questions and lively discussion. And the EACR for organizing it.
Well, we thank you for your availability, and many thanks to everyone who attended. I hope to meet some of you at the Congress in June. Have a nice day. Bye-bye.
Thank you.
Bye-bye.
Bye-bye.
Bye.
Bye.